It was inevitable with the expanded use of immunoassays to discover, detect, and quantitate proteins that demand would ensue for enhanced sensitivity, precision, reliability, and ease of assay development. This webinar will address those requirements as well as the increased performance possible with novel systems. I'm Tamlin Oliver, Managing Editor of Genetic Engineering and Biotechnology News, and I'll be the moderator for this webinar. Thanks for joining us today. Also joining me today are Joseph Barco, Senior Product Manager at Singulex, Andrew McIntyre, Director of Research and Development at Pacific Biomarkers, Sarah Christensen, Research Associate at Pacific Biomarkers, and Phyllis Conliffe, Executive Director of Avanza Pharmaceutical Services, Smithers Avanza. Our four speakers today have a very full program to present, so we are going to jump right in. Joseph Barco will get the presentation started. He will talk about the advantages of ultrasensitive immunoassays as well as the benefits of single-molecule counting technology. But before I get started, I want to remind you we will have a Q&A session at the end of the presentations and encourage you to submit questions for our panelists. We'll answer as many questions as we can. Simply type your question into the Ask a Question box and hit Submit. Okay, Joseph, now you can get started.
Thank you, Tammy, and thank you all for joining us today. I'd like to introduce you to single-molecule counting and speak about how it enables Singulex and our clients to advance life science research. Now, Singulex has three distinct business units that utilize single-molecule counting technology. We have a CLIA CAP-certified laboratory, which provides physicians with data on cardiovascular and inflammation biomarkers, such as cardiac troponin, endothelin, TNF-alpha, and IL-17A. This allows physicians to assess patient risk for development of cardiovascular disease. We're currently developing a new automated clinical diagnostics instrument which will enable hospitals globally to utilize SMC technology to similar ends. This work I'll speak about today is how we use SMC technology in the life science division for research applications in identifying new biomarkers or exploring applications for existing biomarkers. Now, there's a significant need for better quantification for biomarkers and biotherapeutics. Traditional ELISA technology has existed for over 50 years. One of the reasons it has persisted as a technique for so long is because it is specific to the target, it is sensitive enough where it can usually determine if the marker is elevated, and it has a quantitative dynamic range that can often help determine the relevance of following the marker. It has been well utilized in samples of limited complexity, such as tissue culture matrices, with limited requirements for accuracy, precision, and dynamic range, and with the caveat that many samples may be below the detection limit of the assay. Of course, there is an ever-present need for greater sensitivity due to the discovery of newer biomarkers that may not be as abundant as previously characterized biomarkers. You cannot use what you cannot measure, and there's an unmet need for identifying both endogenous levels of many circulating biomarkers as well as monitoring rising levels of a biomarker, which may have implications for the individual that would be otherwise undetectable in a traditional ELISA approach. If an analyte is undetectable or non-quantifiable, it is difficult or impossible to fully assess the individual's risk of association with that biomarker. By way of illustration, is a change in biomarker level a dolphin and relatively harmless, or is it a shark, and do I need to take precautions? To fulfill this unmet need, Singulex and our clients utilize our proprietary single-molecule counting technology, which provides ultra-quantitative data for biomarker or biotherapeutic levels, utilizing a modified immunoassay protocol that is instantly familiar to scientists who have run basic ELISAs or advanced immunoassay platforms. The ultimate goal of SMC technology is to reduce or eliminate ND, that is, non-detectable results. This is a small list of applications that can be improved by eliminating the ND result. When 100% of samples can be quantified, it is easier for researchers to distinguish between endogenous and elevated levels for existing biomarkers. The lower quantitative values enable applications such as patient population stratification for biomarkers, microdosing, or longer time course monitoring for PK and biotherapeutic safety assessment. Singulex and many of our customers have utilized the quantitation improvements to both discover new biomarkers and to uncover new utilities for existing biomarkers. Before I go on, I'd like to distinguish between the terms sensitivity and quantification. I will generally use the term quantification. Throughout the talk, I will use LOQ or LLOQ, which refers to the lower limit of quantification. This is a value that is different than sensitivity. When I say an assay is quantitative at a lower value, I am also tying that value to an expected level of precision and accuracy. I should expect that a sample at a given LOQ is reproducible. Quantification is different than sensitivity. The limit of detection, or LOD, is a signal value that is a certain number of standard deviations above the background. Samples that are measured at the LOD are not reproducible. Usually, the researcher can identify that an analyte is present above background, but the result is not precise enough to assign a concentration for that value. Throughout the talk, I will be referring almost exclusively to quantification values, or LOQ. This is a higher bar and a more robust number than LOD. At this point, I should explain how SMC technology works. At the outset, the steps are identical to a traditional ELISA, or for that matter, any other advanced immunoassay platform. A capture antibody is coded to either a plate or a bead. The plate capture is passive absorption. Bead capture is a conjugation. A sample is added to the plate or bead. Sample types can be serum, plasma, or less common matrices such as CSF, BAL, cell lysates, and fine needle aspirates. Samples can be run neat or with a dilution, depending on the analyte and the required range. Sample is added to the capture antibody, incubated, and then washed. Detection antibody, usually directly conjugated to an elixifluor tag, is then added to the sample with a further incubation and a second wash. Thus far, this is the same as nearly every immunoassay platform in both process and time. At this point, the SMC protocol differs from other platforms. Where other platforms will use this as their assay plate and either directly read the beads or the plate, Singulex adds an elution step. This separates the detection antibody from the reaction. This eluate is then transferred to another plate that contains a neutralization buffer. This mixture is then aspirated into the detection system called the Erenna platform. The Erenna is a flow-based system. The sample runs through a narrow capillary. The detection occurs in a 5 by 10 micron window with a confocal laser focused at that point. The laser excites the fluorophore tag on the detection antibody and reports detected events. The advantages in this detection scheme. First, our entire well can be concentrated down to a smaller volume of elution buffer. As a result, we can quantify, even in situations where there were few analytes present and therefore less detection antibody. Second, we are separating the detection antibody out of the assay plate. Any nonspecific binding or other matrix interferences that may have stuck to the plate or bead are eliminated. This greatly lowers the background of the assay. Our signal is specific to the tag since we are only exciting the fluorophore. There is no enzymatic reaction to monitor or second read buffer to add. This gives us a higher degree of precision that might not have been possible with other platforms, especially when comparing results plate to plate or operator to operator. We have a broad dynamic range as our detection method and algorithm allows us to accurately quantify at the upper and lower end of the curve. More on that in a moment. Because of that flexibility in our detection method, we can report 3 signal modes: DE, or detected events, which are individual molecules passing through the beam; EP, or event photons, which is an intermediate count of photons; and TP, or total photons. These values are used by the instrument software, SGX-Link, to back calculate the analyte concentrations from the photons counted as the sample passes through the beam. The quantification improvements by this method enable better measurements of low abundance biomarkers. The improved precision at low volumes changes the way that biomarkers can be measured. No longer is ND, or non-detectable, an acceptable answer. With that improved precision, the meaning of the numbers reported back on an analyte can be used to understand the clinical relevance of what was otherwise not quantifiable or a less precise measurement. Better quantification makes some assays better or great, even in some cases improved by orders of magnitude. Some of that improvement can be used for reducing the sample requirements for the assay. We have observed in many preclinical assays the sample can be reduced to under 25 microliters per well. And in human samples, assays can be run between 10 and 100 microliters of sample per well and still report quantifiable values. Let me illustrate these advantages in a bit more detail. Here we have an ELISA curve with a limited LOQ of about 10 picograms per mil and a limited dynamic range of about 2 logs. We improve the quantification at the lower end because we can individually count detection molecules passing the laser. Background is reduced in part because we've removed the detection from the interferences that may be left present in the assay plate. In this particular case, we can reduce LOQ by 3 orders of magnitude to 0.01 picogram per mil. At the upper end, our algorithm comes into play. In samples with a high abundance of the marker, the detection antibodies are, of course, not passing through the beam one at a time. Instead, the detector is reading a constant level of signal as multiple antibodies pass through at once. Our detection antibody can distinguish this scenario from the first case and correctly interpolate a value for that sample. Now, I've stated a few times so far that I expect that this will be more quantitative than an ELISA. Now, I'll compare an ELISA with 2 formats that I can use with SMC technology. I'm going to compare a plate-based SMC format, illustrated in the middle, with a bead-based SMC format, illustrated on the right. Our expectation is that the bead format will be the most quantitative. The primary reasons are that I will do a solution-based capture of my analyte and utilize SMC counting for my detection, including using the ELISA fluoroconjugation of the detection antibody instead of the more traditional horseradish peroxidase ELISA chemistry. However, I can also directly convert the ELISA into a plate-based assay and swap the detection chemistry with the ELISA fluor method. This way, I can utilize SMC technology, and I should expect to see an improvement in quantification based solely on that change. So this begs the question, if a bead-based assay is the most quantitative format, why would I not go directly to that format? So the reason for that is that in addition to improving the quantification, the complexity of the protocol increases. If I have a bead-based assay, I need to take more care in washing and perform those steps slower in order to ensure I've allowed adequate time for the beads to settle. Second, the plate as a surface is less expensive than beads as a surface. If both results are better than an ELISA, I may be able to find a point where I can still improve my assay without greatly affecting the cost of the assay. For the data on the next slides, we focused on using mouse IL-17A as a test case and chose antibodies from R&D systems. We chose this because it is aligned with reasons where researchers choose SMC technology. Namely, endogenous sample levels are undetectable by traditional means and sample volume is limited. In this case, we use mouse serum as our sample matrix. First, to establish a baseline, we ran the ELISA as directed by R&D systems protocols. By running at the recommended sample volume of 100 microliters per well, we were unable to quantify values for any samples. We estimated the LLOQ of the assay at 62.5 picograms per mil, based on the precision of the standard curve at that point. Now, at this point, we wanted to make a few comparisons. First, we wanted to set up the assay to work with SMC detection methods. For this, we made 2 changes. We wanted to compare directly labeling the antibody with the ELISA fluor tag and compare that with the indirect method, which is to use ELISA fluor streptavidin plus the biotinylated detection antibody. The latter is the more direct comparison with the ELISA. We also wanted to compare a diluent that we use in our laboratory with the diluent used in the R&D systems assay. Other than those changes, we kept conditions constant for the plate-based assay. We chose 50 microliters sample volume and performed a dilution series for those samples to determine if we could quantify at lower sample volumes. We kept capture and detection antibody concentrations constant between the Singulex plate-based assay and the R&D systems ELISA, so as not to introduce additional biases in the results. This is a result of those changes, and needless to say, it looks good. With minimal changes to assay format, quantification improved by 30-fold from the ELISA to 2 picograms per mil. This was accomplished by changing the detection scheme to SMC technology. The diluent change did not impact results in this limited testing. We do expect that that would have an effect as we started to test spike recovery and dilutional linearity as we validated the assay. Because of the quantification improvements, we can reduce the amount of sample to approximately 3 microliters per well. Since serum from mice is clearly a limited supply reagent, this is not an insignificant difference. When we directly labeled the detection antibody, just the last column shown here, there was a further quantification improvement to 1 picogram per mil. I'd like to point out that the quantification improvements enabled all samples to be above the LOQ. Where my first assay provided no data at all for my samples, this new format provided data on all of my samples. We then did further comparisons with our bead-based format. Building off of the previous assay format, we used the directly conjugated ELISA fluor detection antibody. We then conjugated the capture antibody to our microparticle beads. With our standard conditions, this uses slightly less antibody per well than the plate-based format. What we observed here was a further improvement in quantification to 0.05 picograms per mil. This is 20 times better than the SMC plate-based format and 1,200 times better than our starting ELISA. This quantification improvement was achieved without having to go back and identify new antibodies, which is oftentimes the only way to improve quantification when moving from traditional ELISAs to other advanced immunoassay platforms. In the previous example, there would be limited utility in going to a bead-based format, since my plate-based format provides all the quantitative data I need for my samples. This is, of course, not going to be the case in every assay. In some cases, I will need the ultimate sensitivity provided by the bead-based assay format. But as you can see, there are significant improvements in quantifiable values simply by changing the detection instrumentation. Now, I'd like to leave you with a cost comparison between these options. We determined that from an operational standpoint, that the SMC technology plate-based assay format was the most cost-effective, even in comparison with the traditional ELISA. This traces to 2 specific items. First, the sample volume requirements are greatly reduced in SMC formats, enabling the cost of the sample to be reduced, or perhaps more sample being utilized or saved for other assays. Second, the detection chemistry for an ELISA ends up being more expensive than a labeling kit to add ELISA fluor to the detection antibody. So, using supplier list prices, it ends up being more cost-effective to run a plate-based SMC assay than an ELISA. And of course, there is the added bonus that I am now producing data with my assay in the SMC format instead of a plate full of results below the quantifiable limit of the assay. As expected, the bead-based assay is the most costly, both due to the extra cost of the capture surface and the extra diluents required to run the assay. In this particular example, there would be no need to go to this format since my SMC plate-based format is providing the required data. In conclusion, I hope I've shown you some advantages for considering SMC technology as your detection method of choice. Taking a very basic working antibody pair, I can improve quantification by orders of magnitude with minimal amounts of format variations. The platform itself is providing the quantification improvements, not switching the antibody pair. While there are both plate-based and bead-based formats available, there may be very good complexity, cost, and quantification reasons to choose one format over the other. In comparison to other immunoassay platforms, the researcher has the flexibility and can choose whether they want a bead-based or plate-based assay. This degree of flexibility is unique to the Erenna instrument and SMC technology. Thank you, and please feel free to send in questions as our webinar continues. Back to you, Tammy.
Andrew and Sarah are next. They will talk about their experiences developing and optimizing assays at specific biomarkers. But before they get started, I want to remind you once again about submitting those questions through the Ask a Question box. Andrew and Sarah, the floor is yours.
Thank you. What I'm going to present today with Sarah is development of the highly sensitive PK assays we've done here at PBI. They are part of our custom assay service, and basically we take these assays from development into sample testing in the clinical environment. The assay development starts with reagents and prototyping. We use the same chemistry as Singulex kitted reagents. The labeling kits are purchased from Singulex, and typically we label 1 milligram of antibody yielding 60 to 80%. We firstly identify best working reagents, either from a current ELISA or transferred ELISA. This is a flow chart of a high-level process we do. Basically, we look at reagents initially, acquire reagents. We also look at instrument testing in this. And then we go into proof of concept experiments, which are very, very early evaluations, look at assay optimization, and then ultimately assay validation, which is driven to an SOP and then clinical sample testing. In the early stages of assay development, we look at evaluation. So what is expected values? Normally this is from PK data, and we make an assessment of what is the range of quantitation. We look at assays available, either a current assay or an assay we have developed as an ELISA initially, and we're looking at a limited assessment in this evaluation on the Erenna platform. We look at standard curve, range of quantitation, and we are considering the matrix QC strategy. And we aim to get basic levels of performance from a standard curve up to short-term stability. So what is the assay development process we're using? We look at reagent development, obviously prototyping the assays, then we're moving forward to a verified assay, ultimately a validated assay that's available for either transfer or clinical testing within PBI. And this is supported at PBI by QC and also QCs for lot-to-lot requirements. Ultimately, after we've done the initial evaluation period, we go into optimization. And again, we're looking at review of clinical values. What is the expected PK values? What are the Cmax values? And maybe this is taken from early clinical data or more likely preclinical data. We're looking to reach semi-robust conditions of LLOQ and ULOQ, maxing out the EP value. And Sarah will demonstrate later on some of the values we've taken from these three parameters and how we maximize the ROQ at greater than 4 log. We're looking at a matrix strategy and placement of LLOQ, ULOQCs, as well as the other levels, low, medium, and high QCs, and simple sample QCs. Again, Sarah will outline some of the performance we've got for these QCs. We'll look at MRD. Typically, this is neat, and this gives a great sensitivity, but again, there's a volume restriction. And Sarah will outline some of the methods we've done to maximize the sample volumes we get. We'll also look at matrix effect, and then also any optimization that's required. When we have done that, we normally would go to a qualification. This qualification phase will look at multiple pool matrix screening, alternate matrix selection with any potential interferences. We'll do a selectivity, normally at least 10 donors, maybe normal donors. Typically, this is normal donors, but sometimes it's patient populations as well. Linearity will always start from a 10x ULOQ, but maybe even go up to 1,000x ULOQ. And here we'll also pilot QC levels, either multi-donor, single-donor samples, or a combination of both. And the paradigm is to get qualification ranges that we'll use in our validations. We'll also test the dilution schema used for QCs, because this will be what supports ultimately our validation and our sample analysis. And these sample analyses can go on for over 2 years for PK assays. So in validation, it's a straightforward list of parameters. We tend to do a hybrid process that meets both CAP-clear as well as the bioanalytical guidances. And we're cognizant of this in terms of our LTS program that goes up to 24 months. And one of our senior scientists will manage these programs. As you see, they're listed in this slide here. And they'll be managing the validation through this process and ultimately through sample analysis as well. As an overview, we basically take optimal concentrations of capture and detection, normally from a developed ELISA. And this is typically developed in-house as a basic sandwich ELISA, but then we'll transfer that to a plate version on the Singulex Erenna platform. And we're going to assess typical assay performance. We're going to assess curve shape, ULOQ, LOQ, LLRQ, and anchor points. And Sarah will have some excellent data to present on that as well. We start in standard diluent, but we'll obviously go into matrix for this, assessing throughout this period DE, EP, and TP signals, which again, Sarah will highlight some of the differences of paradigms in using these signals. And secondly, when we're running the standard curve, we're optimizing both the capture and detection, initially in standard diluent, but then also in matrix. We'll also assess the type of plate to use to get optimal plate design and plate assay. And we have an option at this point of getting a very much more sensitive assay, because we can take these plate-based assays and turn them into microparticle assays if needed. The labeling is very simple. It's Alexa floor, and this can be done at specific biomarkers or by the client. Ultimately, the design of these experiments are based on accuracy and precision, and these data will be presented by Sarah latterly through the developments and different paradigms we've used for various assays we've had. We use 3 to 6 levels of control, classically low, medium, high, but sometimes we'll have ULOQ controls as well as sample controls as well. And validation samples are normally at 6 levels of control. And our goal is to get the smallest volume with the highest sensitivity, expand the LLQ from the ELISA to get greater sensitivity, and Sarah will demonstrate some logs of greater sensitivity from the ELISAs we've received. And we also assess triplicate and duplicate analysis to get better curve performance, because our goal is to maximize the plate space, to conserve sample volume, and get curve robustness that will save from plate failures. And with that, I'd like to hand over to Sarah to go over some of the data we've generated.
Thank you. As mentioned before, in assay development, we look at curve shape and the 3 signal types: event photons, total photons, and detected events. We're looking at the log scale comparison, and for our assays, we chose EP signal because it has the widest dynamic range. For early development, we start with a broad range for capture and detection antibody concentration, and then we'll narrow it down to find the optimal concentrations. Typically, we'll run a 3-phase design of experiment. Phase 1, we'll use Singulex standard diluent only. Phase 2, we'll have standard diluent and matrix run in duplicate. Phase 3, we'll run the matrix in triplicate. And I'll show data later comparing duplicate versus triplicate values. Along with comparing curve shape and signal data, it is helpful to compare the data in table format. This table shows 3 different concentrations of capture antibody with the average of 9 curves each for each concentration. Typically, this is also done with detection antibody concentrations, and we do this to compare and find the optimal concentration for both detection and capture antibodies. Another experiment ran for optimizing the assay was well read time. So this figure shows a 384 well plate map showing all 4 quadrants. Each color represents a different quadrant, and each quadrant was assigned a different well read time. This was done to optimize the well read time. Once early development results are compared, the assay parameters are set. And so when we set the assay parameters, we choose a capture and detection antibody concentration. For this one, we chose a 12-point standard curve, and we ran it in duplicate. The top standard was the ULOQ, and it was at 300 nanograms per mil. The LLOQ was 0.59, and then the lowest standard was 0.29 nanograms per mil. And this assay is done neat with no MRD. This is important because the values are taken right from the curve. There's no dilution factor applied. An example of a standard curve performance for the development stage is what I'm going to show next. These standard curves were ran in duplicate. And the initial runs are shown on the left table. So the goal for this, the client wanted an LLOQ of less than 2 nanograms per mil, and they didn't require a dynamic range at 360 is what the initial runs were at. So for the next set of runs on the right table, we reduced the top standard to 300, and also the later runs were better optimized, and we reduced the LLOQ to 0.59. And we were also able to improve the accuracy and precision. Although the accuracy and precision is still kind of wide, it was deemed acceptable for this type of assay based on the client requirements. So another example of assay development results for standard curve performance in plasma is going to be shown in the next section. This is an example of assay run in triplicate. This ELISA assay had an LLOQ reading at 50 nanograms per mil, and their clinical requirement was set at 1 nanogram per mil. So for this one, we optimized the assay. The ULOQ was at 80. The LLOQ we were able to get was 0.078 nanograms per mil, and this is with no MRD, which definitely exceeded their requirements. Once the development phase is complete, we move on to the qualification of the assay. Here are some typical qualification examples for what we run at Pacific Biomarkers. We like to run at least 2 different techs for the assay. Sample volume we'll test, and this is very important for conserving sample. We'll test filtered versus unfiltered matrix, and we can either filter it before use in a big batch, or we can use a 96-well filter plate. And in order to conserve more sample, we recommend not to filter the samples at all. For accuracy and precision, it's always a minimum of 3 runs. selectivity will spike 2 to 3 single donor samples to usually 3 different concentrations. For linearity, as mentioned before, we'll do a 10x ULOQ at least. And then also we'll do a LLOQ determination, and on the next section is an example of that. So for LLOQ testing, we'll dilute a single donor sample down 5 to 6 times lower than the LLOQ of the ELISA assay. Multiple replicates are run, and a power of fit equation is applied to determine where the LLOQ falls at 20% CV. As I mentioned earlier, another assay parameter to measure is sample volume. For this assay, we tested 50, 40, and 30 microliters. The table shows the results for 3 controls, ran over multiple plates with no dilution. To conserve volume, a dilution may be run, but this will cause a loss in sensitivity. For example, if the target is 1 nanogram and a 1 to 10 dilution is performed, it will change the target to 10 nanograms per mil after applying that dilution factor. In the next slide, it'll show QC performance at this optimal volume that we found, 50 microliters. And this was over 80 duplicates, and the accuracy and precision were maintained at less than 20% CV. The previous data tables shown were experiments done in plasma matrix, and the next example I'm going to show was done in serum. So this is typical data for serum standard curve. The ULOQ for this one was 80. There were 2 anchor points below the LLOQ of 0.078. And then you can see the accuracy and precision values there. For early development, it's important to compare different matrix types. A matrix selection is made early on and is based on what the client ideally wants. If the client wants serum, but performance doesn't meet the criteria, an alternative matrix will be tested. The table above compares serum and plasma controls. Each QC was spiked fresh. Later on in qualification, we would re-optimize the assay for the selected matrix and make a bigger batch of controls, enough to last up to 2-year stability time points, as was mentioned before. So moving forward after qualification is validation phase and the invalidation testing. This table shows standard curve performance for a validation. The ULOQ is 100. The LLOQ is 0.39, and the 2 anchor points below. And this was run in duplicate. So the next slide will show some of the QC data that we got for validation runs. The QC data is on the top left is validation runs. The one on the right is in-sample validation, which was run at the same time as the samples. And the table below is overall QC performance. Typically, we'll monitor QC using plus or minus 2 SD. This is based on the cap and clear requirements. And ongoing clinical sample analysis uses the plus or minus 2 SD range.
Thank you, Sarah. So what we've highlighted is basically taking an assay from a basic ELISA through reagent development, prototyping this assay, verifying it, ultimately validating it for clinical testing. And again, we can do these assays for transfer or running in-house. And we offer QC support and the ability to run these assays for up to 2 years with long-term stability fully supported. Thank you.
Thank you, Andrew and Sarah. That presentation was great. If you have any questions for Andrew and Sarah or for Joseph, please do submit them for the Q&A session that will follow Phyllis's presentation. Phyllis is our final speaker. She will discuss assay development and validation in a GLP environment. Phyllis, we're ready for you.
Thank you, Tamlin. Today I will be sharing with you our experience with the adoption and implementation of the Singulex Erenna platform in our GLP CLIA laboratories at Smithers Avanza. Smithers Avanza is comprised of Avanza Toxicology Services and Avanza Pharmaceutical Services. The focus of the toxicology group is mammalian toxicology, and the pharmaceutical division offers bioanalytical services to support biologics and vaccines. This includes assay development, validation, and sample analysis for PK/TK, immunogenicity, biomarkers, and cell-based assays. These assays support preclinical and clinical studies and are performed in our CLIA GLP laboratory. We follow GLPs as set forth in 21 CFR Part 58. By following GLPs, we ensure that our laboratory testing is performed by qualified personnel under appropriate supervision, that we are using validated, calibrated, well-maintained equipment, and also that we are following written SOPs. More importantly, following GLP ensures that all records and events are traceable and reproducible. With recent advances in science, more potent drugs with increased efficacy are being developed. This means we are dosing animals and patients at lower concentrations. Our challenge, therefore, is to measure small changes in biomarker and also to quantitate drugs at levels which were previously undetected. Whereas standard immunoanalytical approaches such as ELISA and ECL could not meet desired sensitivity, the Singulex platform in our hands was capable of detecting drugs and biomarkers in the sub-picogram per mil ranges or lower. At Smithers Avanza, we acquired the Singulex Erenna instrument, and as is required in a GLP environment, we have validated the instrument. Furthermore, as is the case with all of our instruments, we have interfaced the Erenna with the Watson limbs, and this interface is also fully validated. The approach taken to assure that the Singulex Erenna and Watson limbs interface are validated according to our SOPs and as required by the FDA GLP regulations is summarized here in this slide. Test scripts were purchased from Singulex and executed for IQ, OQ, PQ. Working in conjunction with Thermo Fisher, the interface between the Singulex Erenna and Watson limbs was created, and IQ, OQ, PQ performed to test the Watson Singulex interface. This testing ensured that the interface performed as intended from instrument login through data importation and regression in Watson limbs. This interface allows us to set up runs in Watson, export sequence files to the instrument. The sequence files, as shown here, are similar to those generated by the Erenna instrument. The files contain instrument instructions such as number of plates, plate types, read time. Once a plate is read in the Singulex Erenna instrument, raw data from the instrument can be imported into Watson for data regression. The advantages of using Singulex Erenna Watson limbs interface are multifold. However, we've chosen to just summarize the key advantages here. You can import raw data into Watson limbs for data regression. You could select one of the three instrument outputs, that is DE, EP, or TP, as the raw data source. Data regression performed in Watson limbs can be performed using different curve fits such as 4PL, 5PL, plus or minus weighing. Unlike Singulex regression software, you do not need a 12-point curve, and this means that you can have greater sample capacity on the plate. There are other benefits of Watson limbs, such as during development, you can have multiple curves on the plate to determine which curve is best for your method. There's also a full chain of custody of samples. All data processing and reporting performed in Watson is done in a CFR Part 11 compliant and secure environment. Singulex assays, like other immunoassays, require a capture and a detector. Preparation of conjugates, as you heard earlier, is straightforward and is performed in our laboratory. To obtain the highest assay sensitivity, we utilize bead-based Singulex assay formats. Capture, which is attached to the beads, is incubated with the sample, and sample could either be neat or at an MRD. Following incubation and wash step, the detector is added, and then the immunocomplexes are formed on the beads. The resultant detector is eluted from the beads and transferred to 384 well plate where it's neutralized. Prior to the read, the sequence file containing instructions is then imported from Watson to the Erenna, and the raw data is exported from the Erenna to Watson once the plate is read, where the data can then be regressed. As is the case in immunoassay development, there are several critical parameters that are considered during method development. These parameters are no different in Singulex, using the Singulex technology. Some of these are listed here. During development, we look at the concentration of the capture and detector, as both of these reagents are prepared in the Singulex assay buffer. Often, this buffer may need to be optimized in order to reduce the background or increase sensitivity. This may involve changes in salts or detergents. Assays do not have to be run neat, so MRD is also evaluated, and this is done using the standard Singulex diluent to prepare the dilutions. Other parameters that we look at include the analytical range. We look at selectivity, and this confirms the LLOQ. We also look at incubation times during the development. The next two slides show prototypic results during method development. The first one shows conjugate optimization. Under these conditions, the capture concentration was kept constant at 1 microgram per mil, and the detector concentrations were varied at 0.5 and 0.8 microgram per mil. As you can see from this slide, the assay background did not increase significantly with increase of the detector, and the overall performance of both standard curves were quite comparable. This next slide shows the performance of different assay components. Here we're showing the differences in different lots of the Singulex assay buffer using the same capture and detector, and as you could see, there's no lot-to-lot variability that was seen with the change in the Singulex buffers. Likewise, we also looked at different detectors, lots of detectors and captures that were prepared in our laboratories, and again, comparable data were obtained. It has been our experience that the assays on the Singulex platforms are robust and reproducible in different sample matrices as well as with different species. Guidance for the industry, such as FDA, EMEA, together with white papers, set forth expectations for the conduct of large molecule bioanalytical studies in a GLP environment. At Smithers-Avanza, we follow industry and regulatory guidance documents. The parameters shown here in this slide are the ones that we look at during validation of a PK method. These include accuracy and precision, selectivity, dilutional linearity, specificity, and stability. Assay acceptance criteria is usually determined by the inter-assay accuracy and precision. Accuracy and precision requirements as set forth by the white papers and the guidance require that for large molecules that the LLOQ and the ULOQ have a tolerance of plus minus 25% or better, and the LQC, mid-QC, and high QC have a tolerance of 20%. Using the Singulex Erenna platform, we have validated several methods in our laboratories, and it's been our experience that the tolerance and the total error at LLOQ and ULOQ is typically less than 20%, and the LQC, mid-QC, and high QC performance is actually even better, as low as 10% in some cases, with total error being about 20%. And in all of these cases, the LLOQ was less than 1 picogram per mil. Selectivity is the ability of the assay to reliably reproduce results in matrices from one individual to another. Following the guidance, we spike individual samples at LLOQ with the expectation that 80% of the samples would recover at 20% or better. And again, it's been our experience with the validation of Singulex assays that this criteria has been met. Dilutional linearity demonstrates that the sample can be diluted to bring analyte concentrations into the validated range of the assay. Samples at concentration 10 to 100 times higher than the ULOQ have been tested, and we've found that, again, these values do fall well within the standard range of the curve. So in conclusion, at Smithers Avanza, we have fully validated the Singulex instrument along with the interface to Watson LIMS. We have also validated PK methods and discovered that these assays are sensitive with LLOQs less than 1 picogram per mil. The assays are accurate, precise, selective, robust, and reproducible, and moreover, these assays meet the requirements as outlined in guidance documents. Thank you for your time.
Thanks, Phyllis, for that interesting presentation. We're ready for our Q&A session now. We'll get started in a minute, but before we do, I want to remind you about submitting questions for our panelists, and I also want to let you know that a post-webinar survey will deploy shortly in the presentation manager if it hasn't already. You might want to disable your pop-up blockers in order to see the survey, and we hope you do, and that you take a few minutes to respond. Your comments are extremely valuable to us in developing our free webinars. All right, let's get our Q&A discussion started. All 4 panelists are standing by and ready to answer your questions. We have a lot of good questions already, and we'll answer as many as we can. Okay, our first question is for Andrew. Andrew, does detection antibody need to be labeled by Singulex, or can it easily be done in-house with good reproduction?
We found it to be easily labeled in-house. I mean, it can be labeled by Singulex, but we've set up our own process controls internally to manage labeling reagents in-house for PK studies especially.
Okay, thank you. Phyllis, I have a question for you. LIMS import is by using the work list or plate report format? Question mark.
So it is usually done using the work list format, and there is an import option on the analytical actions that allow you to do that.
Okay, Andrew, we are back to you. This is a 2-part question. How do you prepare STD curves in plasma for endogenous analytes, and how do you measure LLOQ for endogenous analytes? Remember, there is no true negative controls.
Oh, that's a great question. For measuring for endogenous analytes within plasma, we would tend to use either a stripped matrix or probably just a standard sample diluent curve, and then look at endogenous QCs. So we manage any endogenous levels by monitoring in the QCs where that was a problem. The interesting second part of the question is how to measure the LLOQ for endogenous analytes. As Sarah showed in one of the presentation slides, we basically just do a dilution series where we make a breakpoint of about 20% CV, and that's how we measure basically the LLOQ for endogenous analytes. And we normally do that with single donors, up to 5 donors in validation to get the LLOQ for endogenous.
Okay, thanks, Andrew. Joseph, I have some questions for you. The first one, are the particles magnetic?
Yes, they're paramagnetic beads.
Okay, that was quick. What is your detection time for LOQ concentration?
So it's a bit analyte dependent. The Erenna will allow you to choose a read time between 15 and 60 seconds per well. But when it comes down to it, the instrument also is smart enough to figure out when it's acquired enough data. So if you hit a certain threshold, it will move on to the next well regardless of whether it's reached that 15 seconds or not.
All right, I have another one for you, Joseph. For plate-based assays, are there specific plates that are required?
No, we use an off-the-shelf NUNC round bottom plate, and we have not extensively tested other plate types. But one thing to keep in mind is that because we're not imaging the plate, you're not tied to using a flat bottom plate as you would be on a normal ELISA. So we actually use a round bottom plate, which allows us to extract more eluate out at the end of the reaction and therefore bring more signal into the machine.
Okay, Phyllis, we're back to you. What's the average number of samples per plate that can be used, and are duplicates or triplicate samples needed?
So on the average, we have about 17 samples on a plate, and the samples are run in triplicates. Obviously, the number of samples can vary depending on the number of standards that are being used to generate the standard curve, but overall it's about 17.
Okay, we're back to you, Joe, and the question is, are there any special sample handling requirements for SMC detection?
In general, no. So some assays will have a minimum required dilution depending on the level of the analyte that's present. Certain samples like urine, in terms of may need like a protease inhibitor or something like that present, as you might do in a normal ELISA. And what we have found in some cases is that there's a benefit to filtering the sample if your analyte level is at or near the LLOQ, and that's because of really of a pipetting precision improvement.
Okay, thank you. Unfortunately, our time is up. Thanks for listening in, and thanks for the great questions. This webinar will be archived for 6 months on our website, www.genengnews.com. If you missed parts of it, you can watch it again, or you can recommend it to friends and colleagues. Once again, I want to thank our panelists as well as our sponsor, Singulex.